Hello everyone and welcome to ITLS Academy Empower the Youth. So today in this lecture we will going to understand the contemporary monitoring methods. So what is contemporary monitoring methods are? So before going into the detail, let us understand why there is a need of such contemporary approach and what are the strategies for food safety by using this method. So let us understand that there is a rapid globalization of food production, and there is, since the population is increasing at a very alarming rate, and everyone needs food for their survival. And nowadays, people are becoming so conscious that they want a food that is free from any sort of microbial contamination. So, since as the food production is increasing, there is also an increase of foodborne illness. Means there are different kinds of food that are directly or indirectly affected by microbial pathogen, and it is affecting this foodborne illness or foodborne diseases. It is affecting in the people across the globe, and this foodborne illness has a serious outbreak of foodborne diseases that have been documented every now and then, and this is directly illustrating the public health, and also they have a social significance of these foodborne diseases. So why it is so? So if there is a poor nutrition and a foodborne diseases from various areas, and it is worsening the health in which the poor are the most exposed and the worst suffer. So globally, at least 2.2 million people are killed annually with borne and waterborne diseases, with the 23,000 people are dying in US alone. So there was a data that US alone has a death rate of 23,000 people dying every year. Like among 6 people, one person is always dying with the foodborne diseases. So this food safety is the assurance that food will not cause harm to a consumer, and it is slowly becoming a major criteria for the sustainable development of any country across the globe. So it is being directly associated with a better productivity and livelihood. So this food safety, they are broadly encompasses on the basis of the four areas, that what should be the microbial safety of the food, because the food, it is a major hub for the major occurrence of foodborne pathogens like E. coli is there, Salmonella species, Clostridium, different kinds of pathogenic bacteria are there. Apart from that, what is the chemical safety of the food? Means whether the food is free from antibiotics, or whether the food is free from pesticides, and it is free from heavy metal contamination or not. Okay, now what is the role of the GMPs, and what are the other novel foods like organic foods are there are pre-packed, packaged or frozen or ready to eat meals that are there in the market. So what is the strategies for their shelf life, and what is the general quality of diet? So with the increasing awareness in both the consumer and as well as the industries regarding the importance of food safety, so it is becoming an important area of discussion, that what should be the you can say the contemporary methods in preserving in the food safety or in the food safety. So let us see the introduction part. I have just given you the overview of what we will going to discuss in this lecture. So the introduction says that to ensure the safety of the global food supply, the contemporary methods are needed for a rapid, effective and accurate monitoring of environmental hygiene, identification of biological hazards and assessment of product quality. So it is needed for sure that yes, if you want to make a food product free from any sort of microbial contamination, so you should have a proper checklist, you should have a different kinds of methods, different types of contemporary methods that are needed for the rapid and effective as well as for the accurate monitoring of the hygiene of the food. See, if the food is hygienic, if the food is free from any sort of microbial activity, then there is no foodborne illness. So it is very important for every industries, for every consumer to consume a food that is free from microbial contamination. Now these contemporary methods, they includes the that is ATP, bioluminescence, then it is PCR, that is polymerase chain reaction, then it is a ELISA, that is enzyme linked immunosorbent assay. So we all are aware of all these method, maybe that is ATP, there is PCR and ELISA, because we have done these method or these laboratories experiments in our labs. So let us understand that how these methods are helping the food industries to check the quality of the food, to eliminate the presence of microorganisms. So let's get start with the topic and let's see the bioluminescence method. So the bioluminescence has gained acceptance as a rapid techniques for quickly assessing the cleanliness of food contact surface, and a variety of commercial systems that are available. One way to determine the level of ATP is by measuring the bioluminescence as registered on the illuminator. Bioluminescence is the light emitted from the living organism that have a ability to produce a luciferase enzyme. And these ATP is an immediate provider of free energy, that is meaning that an ATP molecule is consumed soon alter after its formation, and that the energy association with molecule cannot be stored. As a result, that ATP is present only in living cells, so they disappear shortly, that is within 2 hours after the cell's death. Therefore, the presence of ATP can be used as an indication of cell viability. Okay, are you getting this? How it is helping? See, we all know that this ATP, they are only and only present in living cells. Living cells जो live हैं, right? And they have a tendency that these ATP, they disappear within a very short span of time, like within 2 hours they disappear. After the cell death, means after the cell gets dead, they disappear. So therefore the presence of ATP, means if the ATP is present in living cells, so what they do, they are used as an indication, or they are used as an indicator of cell viability. Okay, so the firefly bioluminescence reactions is an energy consuming process, and as in many other biochemical reactions, ATP is a donor of such energy. In addition to ATP, firefly luciferins requires luciferin, molecular oxygen, and magnesium as a substrate. So what it required as a substrate? Molecular oxygen, luciferin, and magnesium as a substrate. Now this first technique, that is ATP bioluminescence assay, it was first developed in year 1960s, and it is used for finding life in space. And who was the person behind this ATP bioluminescence? It is Chappell and Levin in 1968. So 1968 में they have in 90 Chappell and Levin in year 1960s, they first developed, and firstly they are using this technique or method for finding life in space, because they have this ATP has the characteristic that they can be present only in the living cells. So the technique was later adopted for the detection of microorganism in food. Now since it has a I've written here, it has the tendency or it has the capability that the presence of ATP can be used as an indication of cell viability. So after so many years, this technique is used in food industries. So why it is used in the food industries? Because of its ability to detection of microorganism in foods. So now currently ATP's bioluminescence, it is widely used for rapid assessments for of processing conditions and microbial contamination of food. So whenever you want to check, whenever the industry want to check the processing condition, or whenever an industry wants to check the microbial condition contamination of food, or the presence of microorganism in food, so ATP bioluminescence is used. It has also been used for monitoring the CCPs, that is critical control points, during the HACCP, that is a hazard analysis critical control point. And it is widely used in these two management and for monitoring the microorganism in the food. Now comes the one of the most important, that is PCR. So we all are aware with this PCR technique, because in the laboratory we have done PCR uh, experiments in our laboratories for testing different kinds of microorganism, or for the detection of microorganism. In biotechnology lab we have done this experiment of PCR. So let us understand what actually is PCR is. So PCR, firstly PCR, it was first described in year 1971. And but it gains the, you can say, the important importance in the late 1980s and 1990s, and it was used as a diagnostic tool. And after a decade of two, the technique is becoming more popular than ever, and has been used in many disciplines of life sciences. So in the field of food microbiology, this PCR is one of the primarily used tool, and it is used for rapid detection, or you can say it is used for the identification of microorganisms from the food. Apart from this, PCR also detects food allergens by identifying their DNA sequences, and it also offer a high specificity and stability. And it is especially used in the processed food where the proteins may be denatured. Okay, so again, what is PCR? So PCR, it is stands for polymerase chain reaction, and it is a nucleic acid amplification testing procedure that consists of denaturing, renaturing, and elongating and amplifying a short segment of DNA or RNA. So as with the higher plants and animal, the genetic traits of microorganisms, they are determined by an informed stored in their DNA, that is deoxyribonucleic acid. So this information includes genetic determinants that encodes for enzyme and proteins that are involved in metabolic pathways, constituting structures, virulence factors of microbial cell. And this PCR identify a unique region of DNA that is known as template of a target microorganism. So this is again very important, because in interviews, in examination, they can ask you that what is unique in PCR, and why it is so much used in food and industries. So the simple answer is that PCR identify a unique region of DNA, and that is known as template of the target microorganism. Now what are the four steps, oh sorry, what are the three steps of PCR? Because PCR, again, it is widely used in food industries, so we should also be aware of their steps. And what are the steps that are used in the PCR process. So PCR has mainly three steps, that is denaturation, second is annealing, and the third is extending. Okay, so if you see the first step in this double stranded DNA template, okay, this template, it is first heated. I'm just writing it here. In the first step, it is first heated. Okay, after this double stranded DNA get heated, because of this heat, it they get separate into two single strand. Are you seeing this? This is the first double stranded DNA. After the heat is provided, these two strands, they get separated into two single strand, strand 1 and strand 2. Okay, now the second step says annealing. In this step, the temperature is lowered down. Here the heat is provided, so the temperature was high, but here that what it does, it lowers the temperature get lower down. And after the temperature get lower, it enables the DNA primers to attach to a template DNA. As you can see what they are doing, after the temperature get lowered, the DNA primers, they get attached to the template DNA like this, the arrow one, ACTG and CTGA. Now the third step says that again, here temperature was high, in the second step temperature was low, again in the step 3 temperature is high. Okay, the temperature again get raised again, and the new strand of DNA is made by the Taq polymerase enzyme. Which enzyme is used in making the again the double stranded DNA, or you can say a new DNA strand? That is an enzyme, that is Taq polymerase. Okay, so these three stages, they are repeated how many times? 20 to 40 times, and basically they double the number of DNA copies each time, which is called a thermal cycle, and it is carried out by a PCR machine. So it is carried out by the machine that is known no sorry, not PCR machine, I'm really sorry. It is carried out by the machine that is called a thermal cycler, and depending upon the speed of the machine, because machine has a speed, so depending upon the machine speed, the PCR can be performed in under an hour or up to a few hours. So after the PCR has been completed, the method called electrophoresis, we all are aware of that method, it is again used. And after using electrophoresis, it quickly check the quantity as well as the size of DNA fragments that is produced by the PCR process. Again, in the first step, the double stranded DNA get denatured, or they get separated, not denatured, sorry, they get separated, and it separate into two single strand. As you can see in this diagram, here the temperature is very high. Now in the second step, the temperature get lower down, and in this step, DNA primers, they get attached to the template DNA. Now in the third step, again the temperature are raised high, and the new strand of DNA is made. So what does it do? With the help of an enzyme that is known as Taq polymerase. Here what enzyme is used? I'm just mentioning that Taq, Taq polymerase enzyme. Taq polymerase enzyme, it is used in this step, and with the help of this enzyme, the new DNA strand is formed. And these three stages, that is in denaturing, annealing, and extension, so they get repeated 20 to 40 minutes, and at every time the number of DNA get doubles, and in this way you get more copies of DNA. And at the end of this PCR thing, once the PCR get completed, once the after 20 to after some 20 to 40 times के बाद electrophoresis is used to check the quantities and the size of DNA fragment that was produced. So the temperature at the denaturing stage is 94. Again, I am writing it here. Sorry. So if you see here, the temperature is at this stage, the temperature is 94 to 95 degrees centigrade, whereas at annealing the temperature is lower down to 50 to 56 degrees centigrade, and at the extension or extending stage the temperature is again high, that is 72 degrees centigrade, but the maximum temperature is at the stage 1 then as compared to stage 3. Okay, now moving forward, these are certain samples in which PCR inhibitors are used. So the first one is the sample that is dairy sample, and in dairy sample like raw milk or skimmed milk or pasteurized or a dry cheese, dry milk, or the cheese that is dry or soft, in this fat, protein, calcium, chelators, dead cells, they are so these are certain PCR inhibitors. Then for the sample of meat, the different kinds of meat like chicken, like pork, like turkey, beef, so in this the PCR inhibitors are fat, protein and collagen. And in the seafood like fish and salmon, shellfish, in this a sample phenolic aldehyde protein fat. So these are certain PCR inhibitors that are used. So at different industries like dairy, meat or seafood, in this different kinds of you can say PCR inhibitors are used. Now one of the most important thing, that what is the application of PCR in food industries. So let us quickly understand what is the applications of PCR. So first of all, PCR is good in detecting the microorganism. So they because of their detection power, they are used in food industries. Apart from that, it has a huge importance in different kinds of industries or departments like biotechnology, microbiology. Okay, in clinical diagnostics it is used, in CNP genotyping it is used, in GMO analysis, in detection of plant pathogens. So everywhere this PCR has a huge importance. And let us take an example again to understand this. It has a huge, application in detection of different kinds of foodborne pathogen. So the first one is detection, sorry detection of foodborne pathogen. Okay, detection of foodborne pathogens. So this PCR based methods, they are highly effective for identifying the bacterial pathogens, and they have a very faster and they are more reliable than the traditional or the culture based techniques. So for example, real time PCR assay, they are used for salmonella, providing result in less than 2 days. Okay, and if you want to detect salmonella using traditional methods, so they take 5 days for the detection of salmonella. So PCR, again, it is very fast, more convenient and easy to use. And the second one is the detection of, again I'm writing, the second use is detection of allergens. Okay, detection of allergens. Again, PCR detects the food allergens by identifying the specific DNA sequences, or they are also used for offering the high specificity and stability, especially when the processed food is concerned, where the proteins may denatured. So in this way, it is used in detection of allergens. For example, detection of gluten. Nowadays, gluten based foods are more in trends. Even the peanuts and celery in various food items. And one of the very important use of PCR is again the third one, that is the detection of GMO. Okay, GMOs, that is genetically modified organisms. So PCR is preferred method for detecting the GMOs. And why? Because it has a very high sensitivity, it is very specific, and it is even in a very highly processed food it is used. So the real time PCR assay for detecting and quantifying the GMOs in food product, it is very much used. Again, nowadays, as we all are aware, because of one term, that is the food adulteration. Food adulteration. Okay, where we alter the food, where we alter the quality of food. In this also means the PCR based method, it is used in identify the specific DNA sequences from different species to detect the food adulteration. Even it also ensure the authenticity of food product. And now, for the detection of alteration in apricots or safflowers or in saffron, it is highly used. So again, I would say that PCR has certain characteristics. The first characteristics that make it more useful in in different kinds of food industries is it is fast. It is fast, it gives a result very fast. I've already told you, in the detection of salmonella foodborne pathogen, it takes only 2 days to, or less than 2 days to detect the presence of salmonella, whereas the traditional method takes 4 days or 5 days. Second is, it has high specificity. Okay, it has high specificity. It is it is very highly specific, as well as it has a high stability. Not high, it is stable. Okay, now again, high specificity, high and it is very stable. The third quality that make it that make it very useful, that it has a very high sensitivity. Okay, so these all characteristics, or it make it used in different kinds of food industries, whether it's a dairy industries, whether it's of any beverage industries, or any food industries. So in this way, it is very much useful by the industries. So this is the application of PCR in food control. So that brings to the end of the lecture. I hope you have understood the lecture. If you have any doubt, any query, so you can leave your queries in the comment section below. In the next lecture, I'll be answering the queries. Thank you for watching. Please do like, share and subscribe. This is our helpline number, that is 7080833450. So you can simply call on this number and get updated. And yes, don't forget to press the bell icon, because in this food safety officer program, each lecture is linked to one another. So you can go through all of the playlist to have a better understanding of the programs. So thank you once again.